Hello everyone, this is Shomita's classroom. Again, I will start another new topic, that is what is cell division, what is interphase, what is M-phase, and what is the major difference between this all. So it is included in the chapter cell cycle and the cell division. Let's get started. So what is cell division? Cell division means cell arises from the pre-existing cells. Pre-existing cell means the parent cell. Cell comes from parent cell. Now in this cell division, along with the cell division, cell also follows the development phase. The growth of multicellular organism means the cell division seen in case of multicellular organism. Now this process goes throughout the life. The cell division process goes throughout the life. Now next, another subtopic is the what do we mean by cell cycle? Cell cycle, first this process observed by Howard and Bell in the year of 1953, and cell duplicates its genome. In this procedure, cell duplicates its genome. Its genome means the chromosome or the DNA. Now synthesis of other cell contents also follows in this process. Along with that, we will get two daughter cell. Formation of two daughter cell also involves in this cell cycle. Now cell cycle follows two phases. One is interphase, another one is M-phase. Interphase, as the figure shown, that interphase follows. Later on I will discuss what do you mean by the interphase. Interphase G2 followed by G1S and G2 phase, followed by G1S and the G2 phase. M phase is denoted as mitotic phase or the actual division phase, and also involve the cytokinesis. So this total cell cycle have three important phases followed by three important process or phases. One is the this sub-stages of interphase, and or we can say that interphase, and the mitosis, that is actual division phase, and the cytokinesis. So here it is written average duration of 24 hours. What do we mean by that? If M phase lasts for approximately 1 hour, then interphase lasts for 10 to 20 hours. That means most of the time occupied by interphase, and very small portion, very small time will occupied by mitosis or the dividing phase. Here is the interphase. Interphase, this is the cell, and this is the centriole, microtubule, both form centrosome, and this is the nucleus and the nucleolus. This is the chromatin thread, or chromatin thread will make the chromosome. And interphase, what do you mean by interphase? Interphase period between two successive M phase. Period between two successive M phase means the end of one cell division to the beginning of next cell division. This is meaning the between two successive M phase. Now this interphase also known as resting stage or the preparation phase. So as I already discussed, 90 to 96% of total duration of cell cycle occupies by the interphase stage, whereas very small percentage will occupied by the M phase. Next one, sub stages of the interphase. As the figure shown, G1, S and G2 all are included in the sub stages. That is G1 phase, post mitotic. G1 phase denoted as post mitotic gap phase. That means this G1 is the primary growth phase, and in the G1 centriole divides. Centriole divides, what do we mean by the centriole? Centriole means this is called the centriole. Centriole, this centriole only divide. Okay, this centriole is a part of centrosome. And this G1 phase is the synthetic or the synthesis phase. What is the meaning of synthesis? Here is the synthesis of another organelles, not the DNA. So here it is, that's why I written here no change occur in the DNA content. That is synthesis of the major molecules like RNA, protein, lipid and the nucleotide. G0 Another phase is the G0. Here it is mentioned here, G0. Another important phase, it is called quiescent stage or the zero stage. Why? Because here cell neither divides nor synthesizes the organelles but metabolically active. Metabolically active means cell undergoes secretion, attacking pathogens. Okay, this process. Now, here it is written except cancer cell. What do you mean by that? Cancer cell cannot enter G0 stage. So that's why cancer cell followed indefinitely growth. Next one, S phase. S phase means this is the synthesis phase. Synthesis for what? Synthesis for the DNA. Synthesis for the DNA, synthesis for the histone protein followed by the chromosomal material. That means this is the phase for the DNA replication, not for any other substages followed by DNA replication. Next one is the pre-mitotic gap phase. So before starting the M phase, after G2, here is the starting of M phase. After G2, M phase will follow. So before starting the M phase or the mitotic phase, one gap phase is there. Gap between DNA synthesis and next division. Okay, gap between. So this DNA synthesis phase, and before starting that, gap phase is there. Now synthesizing of another molecules also followed by. Another molecules means RNA, protein, mitochondria and other organelles replicate. Chromosomes condense and microtubule begin to assemble to form the spindles. Next one is the M phase. This is the actual cell division. Cell division means the two identical daughter cell. Cell division. After cell division, we get, as the figure shown, two identical daughter cell, identical, genetically identical, as well as the morphologically also identical. Identical means that it is same as parent cell. Now it is called equational division because after this process we will get two identical daughter cell. That's why it is called equational division. Now in case of animal cells, somatic cell will followed by this mitotic phase, whereas in case of meristematic cell, in case of plant cell, meristematic cell undergoes this division. First observed by Strasburger in the year of 1870, and plants in case of plant cell, whereas Fleming in case of animal cell. Next one is the substages of mitosis. Substages of mitosis, first one is the prophase. So after interphase, see the figure shown here. After interphase, prophase, then metaphase, then anaphase, then telophase. So what do we mean by prophase? Prophase, here is two chromatid of, see here in the figure shown here, two chromatid. This green one, blue one, this is the chromatid, and these are the chromosome. Fully the chromosome, and this one, this is one chromatid and this is another chromatid. So both are sister chromatid. Okay, both are in same chromosome, both are sister chromatid. Okay, that's why it is said here sister chromatid. And the sister chromatid are joined. The circular shape is called the centromere. So this centromere is for the joining purpose. It is present for the joining purpose. And next, so in this phase, two chromatid of each chromosome become distinct, and formation of mitotic spindle. Okay, formation of mitotic spindle. And centriole begins to move. See, centriole begins to move. That's why the spindle fiber, formation of spindle fiber. Now disappearance. See the dotted structure. This is the denotion of the disappearance of nucleolus and the nuclear membrane. Now next one is the metaphase stage. Metaphase stage is the attachment of the spindle fiber. Attachment of the spindle fiber, how it is attached. One special structure is there for the attachment of the spindle fiber. What type of structure? This is called kinetochore. Kinetochore means with the centromere, this shaped structure is present. That is called the kinetochore. This kinetochore structure is very much important. If kinetochore is not present, then the chromatid will not present in the metaphase plate. Next one is the anaphase. Anaphase stage. In this stage, the centromere of each chromosome splits into two, so resulting in the daughter chromatids and formation of, so as they are moving in two different poles, so they are forming different alphabetical structure. shape is alphabetical. Either it is V, J, I, and movement of, here is the, see the figure shown, movement of two daughter chromatid to the opposite pole. That means the chromatid in the opposite pole, and there is the starting of the formation of daughter cell. Now the last stage is the telophase. In the telophase, uncoiling of chromosome to form chromatin spindle fibers, astral ray, see, no astral ray, so astral ray disintegrate. Now again reappearance of the nucleolus, nuclear membrane, and new endoplasmic ER. Here it is shown here, no, ER means endoplasmic reticulum, and the Golgi complex reform. Next is the difference. There is the cell cycle and cell division. Cell cycle is the series of periods of cell life, and it is composed of 3 periods: interphase, mitotic division, and the cytokinesis. Cytokinesis is nothing but the division of the cytoplasm. Mitotic division, it is, we can say that karyokinesis, that means the division of the nucleus. Now cytokinesis means the division of the cytoplasm. Next one is the cell division. Cell division is the splitting of cell into 2 daughter cell and occurs in the last 2 periods of the cell cycle, mitotic division and the cytokinesis. Now next one is the interphase and the division between interphase and mitosis. Interphase is the resting stage or the preparation phase, and mitosis is the actual dividing phase, and interphase is the stage between 2 successive M-phase. That means the one cell division to the next new cell division. Mitosis orderly involves the distribution of cell organelles also and the macromolecules also. So here is 2 important questions for you all. Answer in the comment section. What type of cell division produces identical cell? Mitosis, cytokinesis, mitosis, or meiosis? Next one, which phase involves DNA replication and cell growth? S phase, G1 phase, G0 phase, or the interphase? So, thanks for watching.